Our final presenter this morning is Dr. Kelly Caudle. She is the coordinator of the SEAPIC, or the Clinical Pharmacogenetics Implementation Consortium. This is the consortium you've heard some about from previous speakers that provides guidelines that enable translation of the genetic laboratory test results into recommendation actions for a number of different drugs. Kelly has been instrumental in coordinating the guidelines of writing of the committees that take part in this. She's taken part in the guideline reviews and the writing of the manuscripts, and furthermore, she is involved in clinical implementation of pharmacogenetics at St. Jude's Children's Hospital. She started her faculty career at the University of Tennessee Science Center. She is currently where she received her PharmD and PhD, and she is currently assistant professor at the University of Tennessee Health Sciences Center. Please welcome Kelly Caudle.
So I want to start the presentation today by asking the audience a question. So I'm going to read through the question and the answers, and then we'll go back through and you can raise your hand with your answer. So what do you think is the most challenging aspect of the implementation of pharmacogenetics into the clinic? Do you think it's the translation of genetic information into clinical action, the test cost, test reimbursement, or other economic issues? Is it the availability of high-quality genotyping tests? Is it electronic medical record use, such as the application of clinical decision support? Or do you think it's the clinician and patient resistance and or ethical concerns? So show of hands, who thinks it's the translation of genetic information into clinical action? Good. A good majority of the room, that's good. Test cost, test reimbursement, or other ethical, economic issues? Okay, a few less, but quite a few. Availability of high-quality genotyping tests. One hand, that's good. Electronic medical record use, such as the application of clinical decision support. Good, okay, just a handful of hands. And then clinician and patient resistance and or ethical concerns. Okay, so I'd say about 20% on that one. About 25% of the audience, I think, put this. Is there a pointer? Let's see. We'll put the test cost, test reimbursement, or other economic issues, and I'll let you know I will not be talking about that today. But I will focus on this translation of the genetic information into clinical action. I believe there's a speaker later on the day that will talk about cost. So we gave this actual survey to ASCPT members back in 2010, and 95% of the respondents said the process required to translate this genetic information into clinical action, with the next two responses being that genotype test interpretation, and then providing recommendations for selecting the gene-drug pairs to implement, or really, how do you prioritize which gene-drug pairs to start with when you're starting to implement these into your clinic. So SEAPIC was formed in 2011, well 2009, but our first guideline was published in 2011. And so what SEAPIC does is the Clinical Pharmacogenetics Implementation Consortium, and otherwise SEAPIC, so I'll say SEAPIC the rest of the time. But we have guidelines that are designed to help clinicians understand how available genetic test results should be used to optimize drug therapy, but not whether tests should be ordered. And we take this thinking, this one big key assumption is that there'll be clinical high throughput and preemptive genotyping will become more widespread, and that clinicians will be faced with patients having genotypes available even if they did not order the test with the drug in mind. So I think the great example is many of you today have now got your genotype result, but I have a feeling that if you take it to your physician, he might not know what to do with this information. So the good thing to do is you could also take the SEAPIC guideline with you. Okay, so patients do have this information now. They are taking it to their clinicians. And then you have things like 23andMe that are also providing these types of results with not much interpretation on what you should do with this genotype result. So just a few key points about a SEAPIC guideline. These are really based again on that assumption that the test results are in hand and not to discuss the merits of doing the test. We have a standardized format, so I'm going to kind of walk you through the standardized format of the SEAPIC guideline, but when you pick up, we have now 17 guidelines, so each guideline looks the same information's in the same section. So once you familiarize yourself with one guideline, you should be able to look at and interpret another guideline. We do use scales for grading the evidence and the level of the recommendation. These are peer-reviewed. We have over 160 CPIC members, so this goes through first the CPIC review process, and of course then it goes through the journal review process because they are all published. These are freely available, and I'll show you some examples of places where you can get these guidelines. They are updated. We do update when we feel like there's a change in the recommendation. So there's been new evidence or new studies that have been published that might change what our recommendation is. That's when we would update that guideline. We do have a strong policy on conflict of interest. We want you to trust our guidelines. And as Dr. Mueller told you, that they do closely follow the IOM practices or standards for writing trustworthy guidelines. And we did publish this. There's a nice table in there that aligns our process for the CPIC guideline development with those IOM standards. So as I said, we have over 160 members, and you can see that they're all throughout the United States, and we have several in Europe, in Asia, even some in Australia. So throughout the world, so we are an international consortium. We have members from 86 institutions and I think 16 countries last time I counted. So we really do represent, I think, what's going on in the therapy throughout the world. To date, we have 17 CPIC guidelines with 4 that we're working on currently. I'll just quickly walk you through these. Our first guideline was TPMT and thiopurines. It has been updated, so the parentheses indicates that we have updated that guideline. Also a guideline, as you've seen some examples today, for CYP2C19 clopidogrel, and of course for CYP2C9, VCOR-C1, and warfarin. And I will tell you, we are in the process of updating that guideline, taking into account those utility trials that have recently been published. So in 2012, we had the CYP2D6 encoding guideline published. Also, it has been updated. HLA-B abacavir, SLCL1B1 simvastatin. We also have guidelines for other HLA-B, such as HLA-B allopurinol and HLA-B and carbamazepine, and phenytoin as well. So we also, as we said, we have several guidelines, or 2 guidelines that focus on antidepressants, the TCAs and the SSRIs, and then we have some cancer-related guidelines, DPYD for 5FU, and then of course that TPMT thiopurine. In 2014, we did a guideline on IL-28B and PEG interferon alpha, which is used to treat, of course, hepatitis, CFTR, ivacaftor, G6PD and respiricase. Also a guideline for CYP3A5, tacrolimus, and most recently UGT1A1 and atazanavir. So we are working on a CYP2C19 voriconazole guideline. This will be one of our really true first infectious disease guidelines. I'm excited about this one. We have a draft form of it. Hopefully we'll be able to submit this very soon, and hopefully you'll have it in your hands in the next few months. We're also working through the CYP2D6 tamoxifen guideline. We have one that's in draft form for CYP2D6 and ondansetron, and we just recently started one for RYR1 and inhaled anesthetics. So we get a lot of questions about how do we select these guidelines for starting a guideline for, select these gene drug pairs for writing a guideline. So I'm going to walk you through kind of our prioritization, but I'll tell you that this is posted on the CPIC website. I'll show you where this is. So this, of course, is freely available for you to look at. So when you have a gene that's already subject to a CPIC guideline, if you look at the left side of the screen here, it's a priority 1. And this is because it's likely that if we already have a CPIC guideline for that particular gene, it's likely that this gene has been implemented somewhere. And I think physicians and pharmacists and nurses, they all know that, hey, there's a lot of drugs that might be impacted by CYP2D6. And so they might have questions about other genes that might not be in our guideline to date. So we think that that might be a higher priority than maybe some other genes where we haven't already written a CPIC guideline. But of course, the next step here is to really evaluate those alternatives and evidence. So you really have to know, is there an alternative drug you can use? Does it make sense to use alternative dosing for that drug? And then of course, we have to go through the evidence and make sure there's enough evidence that really supports that association. So CPIC has 4 different levels that we put our gene drug pairs into. So we have the CPIC level A or B, which is where there's a prescribing action recommended. We call these actionable, which means you can change your prescribing habit based on that patient's genotype. And it's usually because there's an alternative therapy or dosing are highly likely to be effective and safe. For a CPIC level C, this means there's really no prescribing action recommended. And this could be very useful information as well, because again, you might have a physician ask you, hey, I know that this drug's also impacted by CYP2D6. Is there anything I need to do for this particular drug? And this is generally CPIC level C when really the alternatives are unclear, but testing is quite common. We also have what we call a CPIC level D, and this is just on the pharmgkb.org, and I'll show you a screenshot of that site here in a moment, where we feel there's no prescribing action recommended, and that alternatives are really unclear, the evidence is weak, and testing is rare or non-existent. So I think of these as you've got a lot of genetic testing laboratories that are reporting back other genes, and you might want to know how important is that gene. You could go to our list and you can kind of see if we ranked it an A, B, C, and if we did give it a D, that means we don't think the evidence is quite strong enough yet to move it into clinical practice. On the right-hand side of this figure here, this is really where there's the gene is not yet the subject of a CPIC guideline. We look at, are these actionable in Dutch guidelines? The Dutch working group also has a set of guidelines. Sometimes these are nominated through a CPIC member. They bring those to our attention and say, hey, have you seen the evidence for this gene-drug pair? We also use the PharmGKB annotation levels, and also if it's mentioned in a professional society or guideline, but it's not really actionable. Again, we go through the same degree of evaluation. We evaluate alternatives, evidence, but here we really take into consideration the degree of testing. If the gene is not being tested, then there's no reason for us to write a guideline because the patient would never come to the clinic with that genotype in hand. So this is the CPIC website. Actually, most of the slides that I'm presenting today are actually available there as well. So taking a lot of notes, I think a lot of that's already posted here. So you can find the CPIC guidelines. You see the arrows, the CPIC guidelines and list of CPIC gene drugs. Just click those links up at the top. They're kind of small here. A lot of information out about CPIC. You can also find out if you're a pharmacogenetics expert and you'd like to join CPIC. There's information there about how to join CPIC. And then also these CPIC announcements, which I think are really helpful. It keeps you really, keeps you in the know of everything that's really going on in the pharmacogenetic community. So if you do click on that Guidelines tab, it'll take you to all of our guidelines, and then there'll be a click to the guideline that you see here. You just click on the drug name, and it will take you to PharmGKB. So this is pharmgkb.org. This is a great resource when you're trying to see what type of evidence is out there to support associations with genes and drugs. And so you can see here that all our CPIC guidelines are posted here. The day they are accepted to the journal, we do try to get them posted almost immediately. And so here, you could also go the kind of the back way here. You could go to pharmgkb.org. You could put in simvastatin, and you would in their search engine, and you would get the same type of result here. Or you could put in SLC01B1 for this example, the gene name, and it will bring up all the information of clinical relevance for that particular gene. It's a great site. I won't talk much more about it. I think it takes some time to get used to it. So if you're interested, go play with it. It's got a lot of information in there. So we also have an implementation. This is one of our resources on our sites. We get a lot of questions on who is actually implementing pharmacogenetics into practice. I think we've got 32 sites listed to date. And so you can, like for here, the Cincinnati Children's, thank you. There we go. So if you click on this link here, it actually takes you to their site describing their program. It's very good if you're thinking about implementing to see what other sites have done and how they've actually implemented. All of our guidelines are linked as practice guidelines in PubMed. I'm going to go through these kind of quickly so I don't run out of time. They're also posted free on PubMed Central. If you haven't heard of the NIH's genetic test registry, this is a great place to look to see if there is a genetic test available for your gene of interest. And so we also do have our guidelines linked to that site as well. ASHP, it's a pharmacy organization, is endorsing CPIC guidelines. I think they've endorsed 7 guidelines to date, but they're working to endorse all of our guidelines. We have a lot of external interactions with other groups. ASCPT has also endorsed the CPIC process and how we develop our guidelines. All our guidelines are posted on guidelines.gov, and this is because it meets those standards set out by the IOM. That's what allows us to post to that site. And then, of course, all these other genomic working groups, we definitely work closely with these. So just quickly, I'm going to kind of walk through the uniform elements of a CPIC guideline, of course, introduction, but there is a focused literature review, so I'm going to show you our evidence table. We go through the literature pretty much pulling out any paper that has an association of some type of the genotype with some type of phenotype, either be the adverse reaction, pharmacokinetic relationship, or an outcome relationship. And you can see here we have a fairly well-established process of grading the evidence where really high includes that evidence that's consistent, it has consistent results from well-designed studies. Moderate is where the evidence is sufficient to determine the effects, but the strength of the evidence is limited by number, quality, or consistency. And then we also have this weak category where evidence is insufficient to really assess the effects on health outcomes. And this is generally because of the limited by number. So for very rare variant, sometimes you might only have one patient in a study with that variant. Unfortunately, sometimes it will get that weak category because it's a rare variant. You're not going to find it very often. We have a lot of information about the gene, so you don't have to really be a gene expert to really implement. We're going to provide a lot of this information for you. So here, and I'm just going to point out the genetic test interpretation. So I kind of want to walk you through how to use a CPIC guideline. So you have this table 1 here, and this is how we assign that phenotype based on the patient's diplotype, okay? So let's use star 4 as an example. This is the table 1 from the coding guideline, the CYP2D6 coding guideline. For CYP2D6, we use what's called an activity score to really assign that phenotype. So if you know your patient is a CYP2D6 star 4, and there might be someone in this audience that actually has that genotype, and you can look at this table, you come down here and you would assign it an activity score. So you would take the star 4, 0 plus another star 4, 0, and your activity score overall would be 0. You come up here, look at this table, assign that activity score, and you would know that your patient is a poor metabolizer. For other guidelines where there is no activity score, we would use just really that genotype. So the patient carries 2 no-function alleles, that would then assign them as a poor metabolizer. So it can be done with an activity score with mostly CYP2D6. Other genes, it's really that combination of the function of the 2 alleles. Then that guideline goes into the drug. So this is really where that gene and that drug collide in our guideline. Again, I'm just going to focus here on our table 2. I call this the heart of our guideline. This is the recommendation. So you see here again, this is our coding recommendation, and using that example again for the poor metabolizer. So we know our patient's now a poor metabolizer. If you look here, you see what the recommendation here is, of course, to avoid coding use due to the lack of efficacy. And we do have this classification of the recommendation for coding therapy. Strong, moderate, or optional. We found that all of these were strong based on the evidence because we thought the evidence was a high quality and desirable effects clearly outweigh the undesirable effects. We do have some moderate recommendations where we just felt that there was a close or uncertain balance as to whether the evidence was high quality and the desirable clearly outweigh the undesirable effects. This is sometimes really limited by the number of papers that really support that association. And we also have this optional category. We've only used it, I think, one time in one of our guidelines where we just really felt like the desirable effects of the pharmacogenetic-based dosing are really closely balanced by the undesirable effects, and we're just not really sure what you should do with that information. We have a lot in the supplement, a lot of supplemental tables. I'm not going to bore you with all these, but I'll let you know they're there. So pretty much if you were implementing into clinic, these tables will really help you with that. But I'm going to focus a little bit more of my talk here on these additional supplemental tables, these resources to facilitate the incorporation of pharmacogenetics into the electronic health record with clinical decision support. Because we think this is a barrier, this is something that's difficult for a lot of institutions to really do. It takes a lot of resources and probably a lot of manpower to get this done. So we are trying to provide examples and really tell the reader how to take a CPIC guideline and incorporate it into your electronic health record. And because of this need, we formed the CPIC Informatics Working Group, and this working group is really a forum for discussion and collaboration focused at informatic issues, with really that goal to provide these types of resources that will really help you take our recommendation and put it into your electronic health record. So just to kind of a little bit of background, the two things that we really focus on are these passive versus active. We feel like pharmacogenetic information really must be presented into your electronic health record in two ways. The first way is really the passive, and this is really just your genetic test interpretation. This is taking that diplotype and telling the patient or the clinician what that patient's phenotype is. And then you have the active. We've seen some examples of this today. These are these post-test alerts mostly where the physician has ordered the drug and now they get this alert that says, hey, your patient has a high-risk genotype. You might want to choose an alternative therapy or reduce the dose. So that's really when that gene and that drug collide. And these are important because a genetic test result should last the patient's lifetime, right? This is not, you can't really put a time stamp on these. It needs to be found in the medical record at a discrete time point where the physician always knows where they can go look at it. And then because it could have been 10 years ago that you ordered the test, you need a reminder that, hey, did you remember that your patient is a CYP2D6 poor metabolizer and coding might not be the best option for this patient. So we do provide for passive, this is just a flow chart from our HLA-B, a back of your guideline, and this just really describes to you how to put the actual test into the electronic health record. This consult interpretation, because I think for CYP2D6, there's 6,668 possible diplotype combinations. So there'll be 6,668 different interpretations of that genotype that you need to put into your electronic health record. So this is a very important part and very time consuming. So we are providing this type of detail into our guideline now. It's also important for you to note in the electronic health record if it's a priority result. Is this high risk? Will this change prescribing for any drug? And that's really what makes it a priority result. And so we do provide this. This is our example from the HLA-B. You have here that if the patient's negative and the consult that would go along with that, and then also if your patient's positive and what type of consult or really interpretation of that genetic test result that goes there. So I'm showing you here, this is what it looks like at St. Jude in our electronic health record. This is the example, I think, for TPMT. So you can see here that this patient's genotype result, it is an abnormal priority, so it is flagged as red. You click there and you get that diplotype, phenotype specific result here. So for active, we also have the same type of figure in the guideline. It kind of really walks you through. This is the example from our SLCO1B1 simvastatin guideline. Somebody's ordered the simvastatin. The computer really asks itself, is there already a genetic test on file? If there is it a priority result? And if it is, that post-test alert would fire. And we also do provide some examples of what that post-test alert should say for your intermediate and for your low function SLCO1B1. So this is how we do it at St. Jude. We actually put we put it into the problem list if they have a high risk genotype. So you can see here this patient has a CYP2C19 ultra rapid metabolizer, and they're also SLCO1B1 intermediate metabolizer. And yeah, this problem list entry really serves as that discrete data element for interrupted point of care, because again, if you put it into one that's time stamped, you have to search back 10 years to find that result, and that's just not adequate. So this is what one of our post-test alerts look like. Here you've got, for TPMT, the physician went and ordered TPMT. It found out that his patient was a TPMT intermediate, had intermediate TPMT activity, and you were supposed to consider a lower dose here. And what's I think really nice is that the clinician can then choose to dose this right from this screen. They can modify it, they can cancel the order, or they can actually prescribe the recommended dose. So as I said, a little bit about the translation table. So this translation table, you'll hear this term a lot, is really taking that diplotype to that phenotype. And again, for, let's see, I think CYP2C19, there was 596 possible combinations, many more with CYP2D6, as I've already discussed. So these tables are provided as a link out from our guidelines. So this is really the first step when you're implementing. This is the first thing you have to do. You have to assign this phenotype. We've done it for you here. This is also a nice table here. This is more of a allele definition table here. So as I said before, it really is that combination of the allele functions that really dictate that phenotype. So what the SEAPIC guideline does is we go through the literature and we really try to be very precise in how we assign that function of that allele, because it becomes very important when you're assigning that phenotype. And so am I out of time? I'm good. Okay. All right. Then I'm going to keep talking just a little bit because there's this project and we just finished it up. It was just accepted into genetics and medicine. And one thing that as we were putting these resources together, one thing that really stuck out is that laboratories, our guidelines, patients and physicians were not using the same terms to describe phenotype or this allele functional status. And this is really important, especially when one computer is trying to talk to another computer. They need to be using the same language for them to understand. And so what we did was we had this project where we sought to standardize the phenotype terms in the SEAPIC guidelines and to harmonize these terms with external groups. So we were working with ClinGen and IOM and some other groups. But the two types of terms that we were looking at were this allele function status and also these phenotype terms. And so I think for your phenotype terms, you guys are used to hearing ultra-rapid metabolizer, extensive metabolizer, intermediate metabolizer, and poor metabolizer, right? But I think there is a lot of interest here, not only in standardizing these terms, that we all use these terms, but to make them so that they make sense. I think people hear the term extensive metabolizer and they think they're more of a rapid metabolizer. And then you have to explain to the patient, no, actually that means you're normal. So why not just say normal, right? So we had a lot of discussion on not only just trying to standardize these terms, but also trying to make them so that they were reasonable to the clinicians and to the patients. So we did what was called a modified Delphi process. I'm not going to really walk you through this whole thing, but it's really where you start. And please, you don't have to read this slide. I know this violates every rule to slide making known to man, but that was the point. So you could see how many terms that we started with. So this is showing you phenotype terms, and you can see that for all these different genes, we started out with quite a few genes. I surveyed pharmacogenetic testing laboratories and asked them to send me what terms they were currently using. We also went through the literature and pulled out any terms that were currently being used in the literature. And we got quite a few of terms back. So the first round was really just used to get rid of the bad terms people didn't like. So we had an expert panel. I think we had about 60 people participate in these surveys. There were 5 rounds of surveys. And we were looking for consensus of 70% or greater, but I will tell you by the end of this process, we did get 90% consensus for the final term. So I'm going to show you these terms, and I ask that you start using these terms. They will be used in SEAPIC guidelines, and then we do hope these terms have been endorsed by AMP, so your pathology group, so the genetic testing laboratories, and we're trying to now send out letters to the laboratories asking them to start using these terms as well. So for allele function, so again, this is just the allele, increased function, normal function, decreased function, no function, and then we also have a term for unknown function and uncertain function. A lot of discussion with the group on do we need 2 separate terms, but it really does seem to matter in cases on what your clinical decision might be. So we do have 2 separate terms for that. But here I want to show you our final phenotype terms. I think these are the most important. These are the ones you're used to looking at. These are the ones you know. So here, this is for your drug metabolizing enzymes. So CYP2C19, TPMT, DPYD, UGT1A1, they are still drug metabolizing enzymes. So all these terms should be used for these as well. So ultra rapid, I think you guys are used to that one, but this rapid metabolizer group, I'm actually going to use it in the voriconazole guideline that's coming up for CYP2C19, but I want to draw your attention to normal metabolizer. It was very apparent very early on in the survey that people did not like the term extensive. It actually fell out, I think, at survey 2. So actually the term we're going to be using now is normal, but I will tell you that there were genetic testing laboratories that were already reporting normal metabolizer and had already quit using extensive metabolizer. So now you just tell your patient they're a normal metabolizer. There was a lot of discussion about this intermediate metabolizer group because it's kind of confusing if it should go between normal and poor or between ultra rapid and rapid. But I think it was such a historical term. The other options no one else was using, so I think the experts really chose in the end that intermediate should stay, but with some type of interpretive language. with that one. We also did this for your phenotype terms for transporters. So now we're saying increased function, normal function, decreased function, and poor function. And then we also have a phenotype term for high-risk genotype. So we used to say carrier, but that's not really right, because in the disease world, if you're a carrier, it just means that you don't have the disease, but you carry that mutation. So it was really wrong, because here these are high risk. If you just carry one, you're at a higher risk for these reactions with these drugs. So it really wasn't the correct term to be using. And so that was one point that that our experts felt very strongly about, and so now we're going to be using positive and negative for these terms. And so that will end with that. I know I went over, so I apologize, but thank you for your attention, and if you have any questions, I'd be happy to answer. I think we're